It's a great pleasure for me to introduce Najoua. She's a great pleasure to work with, and she's a really important part of the lab and has contributed to many projects. But today she's going to talk about the project that she has really driven herself. I think that there are many important messages that we can extract from this talk, and I'll just try and do that very briefly. I think the first thing is that it is really a great story. It's full of unexpected surprises and real translation application. But that story took a very long time and a lot of work to develop. Najoua started in the lab in 2010, and it's only now that we hope to get it accepted in Cancer Cell after we sent back the revisions a couple of weeks ago. So it really did take a long time and a lot of work, and I think that we all need to think about that when we're thinking about the big papers and exciting studies. It took a long time because what was ostensibly a very, very simple question, how does Smac-mimetics induce TNF to kill cancer cells, turned out to be very, very complex and completely the opposite of what we could have anticipated based on an enormous literature. That is part of the reason it took a long time, and I certainly don't wish to spoil Najoua's thunder, so I'm not going to go into the surprises and the twists and turns. So I'll just finish with two observations. I think that for me, the project has really highlighted two very important things. The first is the benefit of discussing our science. As most people here know, I strongly believe that we benefit from communicating our science at the earliest possible stage and getting feedback from a large number of people. And that has really influenced this project, from the division meetings where Davo has made particular comments that have helped to shape the direction, to other people talking like Kurt Lakovich telling us about the fantastic chemical library screen, chemical kinase inhibitor library that was initially set up by Chris Burns. And without those kind of little important moments, the project would be a very different shape to what it is today. And the second thing is that I would like to emphasize, and what was brought home to me, is what a fantastic research environment we have here in wehi, in particular, but also in Melbourne and beyond. So, for example, we were able to immediately apply some of the things that we'd found to models that have been set up by Paul Eckert and Stefan Glaser and Ricky Johnston in the Peter Mac. And then we were also able to access colleagues in the Baker, Andrew Way, to look at human patient samples. And I think that the reason that the project didn't take longer, if I can finish that way, is that we were able to really tap into some fantastic resources. And I'm sure that Najoua will acknowledge them properly at the end of her talk. So with that, I'm very much looking forward to the talk. Thank you very much.
Hello, everybody. So I need to acknowledge I'd like to begin by acknowledging the traditional owners and custodians of the land of which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respect to the elders past and present and the elders from other communities who may be here today. So, okay. This is really strong light. Sorry. Okay, so today I am going to talk about a really unexpected combination treatment that we would like to translate into the clinic, which consists on inhibiting the IAPs and the P38 MAP kinase. So first, just to spare a question from Devo, all the bars are mean, plus minus the SEM, and we've used t-test, and circles are individual data. So you can go back to sleep, Devo. All right, so first, I would like to just start with a really simple, for me, sorry for the non-signaling people, but simple slide on how apoptosis work, and especially because I'd like you to situate my protein of interest, the IAPs. So, as you may know, the cells can die through the intrinsic and the extrinsic pathway. The BCL2 family block the intrinsic pathway, and this is what MGC does. And in our division, we are looking at the role of IAP that block the extrinsic pathway, and we are interested in looking at those small molecules that target those protein and that are now used in treating cancer because they prime cancer cells to apoptosis. So IAP stands for inhibitor of apoptosis, but they have more function than what their name implies. So they indeed inhibit caspases to block apoptosis, but they also keep in check the RIP kinase activities to limit spontaneous necroptosis, that is an alternative cell death independent on the caspase activities, but they also regulate the NF-kappa B and MAP kinase pathway to regulate cell survival and inflammation. So the most most characterized IAP member is X-IAP, and it contains 3 BRR domains that are conserved amongst the IAP members, but it also has a ring domain that confers a E3 ubiquitin ligase activity. So in steady state, X-IAP binds to caspase 9 and caspase 3. But when the cells are stressed, the mitochondrial pathway is activated, and SMAC-Diablo is released and bind to X-IAP, and caspases are released to terminate apoptosis. The other 2 IAPs members that have been well characterized are CIP1 and CIP2, and those are less potent in binding caspases, but they do inhibit caspase 8 through the ring domain. So the way CIPs inhibit caspase 8 activity is through mainly through the TNF signaling. So TNF is the master cytokine in inflammation, and it activates survival pathways that are involved in the transcription of genes that are implicated in inflammation and cell survival. So when TNF binds to its receptor, adaptor proteins are recruited to the membrane, and within this complex, CIPs ubiquitinated RIPK1, and then other E3 ligases come along, and they build up this ubiquitin platform. And this platform serves as docking site for kinases that are recruited and then activate the NF-kappa B to induce the expression gene. So the ubiquitination of RIPK1 stops this protein to be involved in a big cell death platform called ripoptosome. And within the ripoptosome, there is the recruitment of RIPK3, FAD, caspase 8. And there is another level of regulation of the caspase 8 activity is through the expression of C-flip. C-flip is a natural inhibitor of caspase 8, and within the ripoptosome, it directly inhibits caspase 8 activity. So because apoptosis has been often found to be deregulated in cancer, it was obvious to look at the level expression of IAPs. And for example, in liver cancer, they've been found to be, the CIP gene, they've been found to be amplified. In multiple lymphoma, there is a translocation of gene between the MALT gene and the CIP gene that produce this fusion protein that activate the NF-kappa B. And there is a lot of publication that have shown that IAPs expression are elevated in many types of cancer, and their expression has been correlated with chemoresistance, cancer progression, and poor prognosis for patients. So their role in cell death, regulating cell death, and in regulating cancer and inducing cancer chemoresistance has attracted the company to make a molecule that specifically target IAPs. And that has been able when they found out that through the crystal structure of the BIR3 of XIAP and Smac-mimetic, and they found that there is 4 important amino acids that are actually well conserved in amongst the different IAP antagonists. So companies has developed a small molecule that mimic those 4 amino acids, and those drug called now Smac-mimetic because they mimic the binding of Diablo, can bind the BIR3 and 2 of XIP, or the BIR2 of CIP1 and 2 to dimerize those 2 molecules. So how does SMAC kill cancer cell? So, so that has been found by our lab through James Finn's work, but other 2 independent groups has found that when you treat cells with Smac-mimetic, you have secretion of TNF, and this autocrine TNF binds to its receptor, and the cells, they become sensitive to TNF. And it is because RIPK1, which is no longer ubiquitinated, is involved in the riboptosome where the caspasate gets activated. But the level of the caspasate activity is going to determine what type of cell death the cells will undergo. So when the caspasate activity is high enough, within the riboptosome, it cleaves RIPK1 and RIPK3. And it cleaves caspasate to terminate apoptosis. But when the caspasate is low, RIPK1 and RIPK3 are recruiting MLKL to induce the caspase-independent death called necroptosis. So to date, there is a number of Smac-mimetic that have entered the clinic. And amongst the ones that are in clinic, we are working with one of the most advanced one, Birinipan, which has been designed by Tetralogic. And now he just had completed phase 2, and the result has not been yet published. And it has been tested, it has went through phase 1 and has shown that it's well tolerated in human. It has been tested in different types of cancer, ovarian, breast, AML, MDS. And now we know that thanks to the work from Mark and Greg, it also tested for another type of disease, which is HBV. So, unfortunately, not all cancer cell lines are sensitive to Smac-mimetic. And here for example, we screen a number of cancer cell lines for their sensitivity to Smac-mimetic and to TNF. And you can see that all those cell lines did not die. But when we added TNF on top of Smac-mimetic, they all been sensitized to Smac-mimetic killing. So that suggested that the lack of TNF secretion in one of the major molecular mechanisms of resistance to Smac-mimetic toxicity. So when I started my postdoc, that was my duty, is to answer how, when we treat Smac-mimetic, TNF gets secreted. So we already had some clue through James Finn's work, when he found that cells that are treated with compound A Smac-mimetic increase TNF secretion, but when he blocked NF-kappa B using an I-kappa B super receptor, repressor, the TNF secretion was completely inhibited. So that suggested that the NF-kappa B was responsible in the TNF secretion induced by Smac-mimetic. So then Ling Wang, she was also interested in this question, and what she found is In macrophage, when she treated with Smac-mimetic compound A, she found that TNF was highly secreted in the supernatant. But when she inhibited RIPK1 using necrostatin, there was a complete inhibition of TNF and Smac-mimetic induced TNF. And similarly, when RIPK3 was absent, there was an inhibition of the TNF induced by Smac-mimetic. So the first hypothesis was that because RIPK1 and RIPK3 are involved in necroptosis, and at that time necroptosis was already thought to be an inflammatory cell type because it induced the release of DEMS, and DEMS are known to be involved in cytokine, pro-inflammatory cytokines such as TNF. So we thought that it was necroptosis induced by Smac-mimetic that induces the TNF. But actually, when I tested the MLK knockout, there was no change in the TNF secretion. So that suggested that the role of RIPK1 and RIPK3 in regulating TNF was independent from their role that they had in inducing necroptosis. So here we found 3 positive regulator of TNF secretion when we treated cells with Smac-mimetic. So then we refined our question, and actually we thought that how if we could find the negative regulator of TNF secretion upon Smac-mimetic, and that we could target those negative regulators to be able to increase the TNF secretion and to be able to increase Smac-mimetic killing. So to address this question, I've used bone marrow-derived macrophages because we knew that they were inducing TNF upon Smac-mimetic. And I screened for kinases that would be involved in the TNF secretion induced by Smac-mimetic using kinase inhibitor library. So this library was contained inhibitor that was targeting different signaling pathway, and it was including preclinical as well as clinical compounds. So here is a heat map of showing the level of TNF secreted by macrophages on the top after treatment with the different kinase inhibitor, and here with Smac-mimetic. So you can see that none of the kinase inhibitor induced TNF on their own compared to the positive control. But we have a differential response when we added Smac-mimetic, and for example, the kinase inhibitor that targeted the JAK kinase give a mixed response. Some were inhibiting the TNF, others were increasing, and others probably were not changing much. Similarly, the kinase inhibitor that targeted the PI3K AKT pathway was giving differential responses. So one family of kinase inhibitor was quite interesting because the ER1 and 2 has been shown to be overexpressed in cancer, and actually there is a lot of compounds that are tested in the clinic to treat cancer. And here, I would say that most of them were increasing the TNF induced by Smac-mimetic. But we prefer to work on more interesting results in our lab. So we went to look at the kinases that targeted P38. So here it's the TNF secretion of 12 P38 inhibitor with Smac-mimetic, and amongst the 12, only 1 did not induce TNF. So we were happy, but then when you start to go on PubMed and look at the role of P38, you found out that it is the most characterized and studied kinase in the world. And then you don't want to read all of that, but you want to So you want to know what is the role of P38 in the TNF secretion. And again, quite few papers showing the role of P38. And what those papers said, I didn't read them, but let's say I should say. But they show that P38 is a positive regulator of TNF, and upon different cytokine, upon toll-like receptor, and upon stress. So actually, P38 plays a role in TNF production in all steps of the TNF production. It phosphorylates transcription factor to increase the transcription. It phosphorylates MK2 to increase the RNA stability, as well as the translation. So because of its role in regulating TNF, a dozen of companies have developed the P38, and now they are in the clinic. And to date, there is like more than 50 studies that are using P38, and most of them are to reduce the TNF in inflammatory disease. So in the clinic, they are used to reduce the TNF, and in our screen, we found that they increase the TNF secretion. So we were quite puzzled with these results. So the first thing that we thought, it was like, okay, this is an off-target, and the P38 that we're playing with are not great. So we checked whether they were doing a good job, and we looked at their ability on inhibiting TNF when we activated the TLR. So here it's the TNF production in macrophages, BMDMs, and you can see that LPS that activate the TLR4 increase the TNF secretion. But when we inhibited P38, it nicely decreased the TNF secretion. So we found that with this P38, we found that with another 3 or 4 P38. So our P38, they were doing what they were supposed to do, what the 9000 paper was saying. But when we treated with Smac-mimetic, we had the exact opposite. And so then we said, okay, that's something else. Let's try the MK2 inhibitor that is always also a really well-characterized kinase to be involved in TNF secretion. And again, we have the exact results. MK2 inhibitor inhibited the TNF production induced by LPS, but it induced the TNF when cells were challenged with Smac-mimetic. So we're like, okay, probably it's not an off-target. There is no off-target when cells are treated with TLRs, but for some reason, upon Smac-mimetic, we could imagine that the 11 P38 that we've tested was targeting another XYZ protein that upon Smac-mimetic. So to close this chapter, we just looked at the knockout cells. And what we found is cells that were lacking either P38 or MK2 had a greater amount of TNF upon Smac-mimetic. So we are on target, and that's great, but also we are showing a real paradox where it looked like when we treated with Smac-mimetic, the P38 pathway was doing the opposite of what it was supposed to do in regulating the TNF secretion. So then we wanted to know what was how P38 and MK2 limit the TNF secretion induced by Smac-mimetic. So is it through an increase of the transcription, or is it through an increase of the RNA stability? So for that, we looked at level of primary RNA that contain all the introns and other stuff. And we also looked at the mature TNF RNA. So what we found is cells that were lacking P38 of MK2, they had an increase on the primary RNA as well as the mature RNA. And this result suggests that inhibition of P38 and MK2 increased the transcription of TNF upon Smac-mimetic. So we don't have any data to rule out or not the role of this pathway in increasing the RNA stability. But at least we know that it's increasing the transcription. Okay, so now what we want to know what is driving the TNF production when we inhibit IAPs and P38. And as I mentioned, P38 is one of the main pathways that regulate TNF. So when he's not there, so who is doing the job? And a TNF gene is tightly regulated by many other pathways. And here we know that the NF-kappa B pathway is directly involved in the transcription of TNF, and the other two subgroups of MAP kinase, ERK and JNK, they also involve in increasing the transcription as well as the RNA stability of TNF during inflammatory process. So what we did is we looked at the phosphorylation events in cells lacking P38 upon Smac-mimetic. So here it's wild-type BMDMs. When they're treated with Smac-mimetic, you have increase of P65, but loss of P38 did not significantly change the phosphorylation. What we found instead is deletion of P38 increased phosphorylation of JNK1 and 2, as well as ERK1 and 2, when cells were treated with Smac-mimetic. We had more profound results when we looked at the MK2 cells knockout. And here you can see that, again, there is no significant change in phosphorylation of P65 between the wild type and the MK2 knockout. But if you look at JNK, you have a strong increase and even stronger at the phosphorylation of ERK1 and 2. And we always like to make sure that our inhibitors are doing what we found in the knockout. And again, similarly, cells treated with a P38 or MK2 inhibitor did not change the phosphorylation of P65, but increased the phosphorylation of JNK and ERK upon Smac-mimetic. So here we're showing that when P38 is not here, we have an increase in the phosphorylation of JNK and phosphorylation of ERK. So are those guys involved in the TNF increase that we've seen when we treated the cells with SMAC and P38 inhibitor? So looking at the TNF secretion in BMDMs, we found that, as previously shown, the treatment with Smac-mimetic increased TNF in the supernatant, but inhibition of P38 increased even more. But when we inhibited JNK with two different inhibitors, we have a complete abolition of TNF secretion induced by the combination. And similarly, here we treated with a two MEK inhibitor that is an upstream kinase of ERK, and we found similar results. So that suggests that, yes, the phosphorylation of ERK and JNK is responsible for the TNF increase when cells are lacking IAPs and P38. So that's what this slide is summarizing. And what we found is, for some reason, when cells are treated with Smac-mimetic, P38 MK2 limit the activity of JNK1 and 2 and ERK1 and 2 to limit the TNF production. But when we apply inhibitor, we unleash this brake and TNF gets more secreted. So our initial question was, will we increase the killing of SMAC if we increase the TNF secretion? And the response is yes, we did. And then what we wanted to know is whether this death was going through an apoptotic pathway or a necroptotic pathway. So we looked at the role of each of those proteins involved on both processes. So here we're looking at the cell death in macrophages. And as I mentioned, we found that macrophages are sensitive to Smac-mimetic compound A. And the inhibition of P38 increased the death induced by Smac-mimetic. When we inhibited RIPK1 using necrostatin, we found a complete inhibition of the death induced by the combination. So because RIPK1 is involved in apoptosis, necroptosis, and in the TNF secretion, we couldn't figure it out what type of death was inhibited in here. So what we did is we looked at the role of RIPK3, MLKL, and caspase. So when RIPK3 was absent, we had a decrease of the cells. But again, RIPK3 being involved in necroptosis and the TNF secretion, we couldn't know what was the role of RIPK3, whether it was through his regulation of TNF or through the regulation of necroptosis. But when you look at MLKL knockout, you don't have any change in the cell death. So that means that actually necroptosis was not required for the killing induced by Smac-mimetic plus P38 inhibitor. And looking at cells that are lacking caspase along with RIPK3, we have a complete inhibition of the cell death. So that suggested that apoptosis was induced when cells were treated with SMAC and P38 inhibitor. So to make sure it was an apoptotic type of death, so we looked at caspase cascade by looking at the cleavage product of caspase, its substrate caspase-3, and the caspase-3 substrate PARP. So here we're looking again at cells that are lacking P38 or MK2. And what we found is after 3 hours of Smac-mimetic treatment, we have an increase of caspase activity as well as PARP. But we found again a more profound effect when MK2 was absent, where caspase was caspase cleavage was increased as well as caspase-3 and PARP. So again, our inhibitor, they're doing a great job like we want. They are also increasing the caspase activity. Here, the P38 and the MK2 strongly increase the caspase and caspase-3 as well as the PARP. So that suggested that killing by Smac-mimetic plus P38 was going through an apoptotic route. So here we demonstrated how SMAC and P38 inhibitor work. And our molecular demonstration has been done all in non-malignant cells, in macrophage. So can we use this combined treatment to treat cancer? And when I started the project, I was using the Smac-mimetic compound A. And compound A is a really good inhibitor of all IAPs, CIP1, 2, and XIP. And what we found is Smac-mimetic that target all IAPs are highly inflammatory because of this high release of TNF, but as well as IL-1 beta, like James Vince has shown. And actually, Lin Wang has confirmed those results using knockout mice, where she found that if you knock out CIP1, CIP2, and XIP only in the myeloid lineage, the mice have a systemic inflammation and they are rented and they don't go over 8 weeks old. So that was showing how specific our Smac-mimetic were in targeting IAPs. So those observations led Tetralogic to develop a new Smac-mimetic called birinipen. And this one is less specific in inhibiting XIP. And that was enough to make this compound well tolerated in mice. But actually, it's also well tolerated in humans because, as I mentioned before, it's actually it's actually trialed in a human clinical trial. I'm sorry. So we've chosen to test our combination in AML, and that was for different reasons. So the first one was because AML are birinapant is tested in AML, in human AML, in phase 2. And the other reason it was from the work that has been conducted by, Gabriella Brumati in our lab, where she made those different type of AMLs, and she tested those leukemia for their responsiveness to birinapant only. And she found that that was a differential response. Here we're looking at cell survival. And for example, the MLL-AF9 and ENL were highly sensitive to birinapant compared to the hoxanized mice-1 and the NUP98. But when she tested in vivo, it turned out that, yes, birinapant was giving a survival advantage, at least for the ENL, but they finally all relapsed. So the mice were dying after we stopped the treatment. And the third reason is this type of AML, unfortunately, has no new therapy for 20 years, more than 20 years. And the only option for patient is intensive chemotherapy. So this type of leukemia is really in a desperate need on finding new therapeutic approaches. AML is a really complex disease. It's complex because it's an association between a cooperation between chromosomal translocation and genetic mutation. So more than 50% of the AML patient, they carry those different chromosomal translocation. And several labs have developed a mouse model of AML. And what they did is they took those translocated genes and they put them into progenitors, and then they put those progenitors into mice, and after a couple of weeks, the mice, they develop an AML. And those models has been quite handy because they reflect the human disease, and they also able to predict the response of the response in the clinic to new therapeutic drugs. So we start off with looking at the leukemias that carried the MLL fusion protein, the MLL, ENL, and AF9, and this translocation is found in approximately 15% of the human patient. So here we're looking at the death induced in vitro, and in blue is cells treated with birinapant only, and you can see that they were relatively resistant, but when we inhibited P38, we have a significant increase in the death induced by birinapant. So next, we wanted to know whether the overexpression of the oncogene RAS would stop the efficiency of the combination. And RAS is found in 40% of its mutated RAS is found overexpressed in 40% of MLL fusion patient. And plus, the other reason was that RAS activate ERK, and we were thinking that could have a potential crosstalk between the RAF-ERK pathway and the P38 pathway. And it turned out that no, RAS did not stop the efficacy of the combination. As you can see, there is a great increase in the cell death induced by birinapant when we added the P38 inhibitor. So we tested another 4 type of AMLs, and we found that, the NUP98 oxanine and the oxanamide1 were also sensitized to the combination, but the CBF MYH11 and the AML-E2 were not responsive to the combination. But it turned out that actually we were able to sensitize all the AML that carried translocation that were giving the poorest prognosis compared to those two that had the best prognosis in the clinic. So then that was in vitro, so you want to know if you can cure mice. But before, we needed to make sure that injecting those two drugs into mice would not kill them, and in healthy mice. And the reason we needed to make sure if it was because here we show that Smac-mimetic that target all IAPs, they are inflammatory because they secrete a lot of TNF. So, but birinapant is not, because by not inhibiting XIAP, there is less TNF and it's well tolerated. But our hypothesis is that birinapant and P38 increase the TNF. So it was possible that putting those two drugs into mice would phenocopy the side effect that we had with compound A. So we wanted to do it, but Tetralogic, they say, no, you do not touch to our clinical compound. You do not play around with that. So you test first another Smac-mimetic, which is also inflammatory, in addition of the P38, and then we discuss. So here I was a little bit nervous because 072 is as good as compound A inhibiting CIP1 and CIP2 and XIP. And Lin Wang, she injected this SMAC into the mice, and she saw that it was inducing an inflammatory phenotype. It was inducing splenomegaly. So the idea was, will the addition of P38 increase TNF, and will it either kill the mice or make the mice really bad that we stop the project? So no, we did not stop the project. It turned out that the mice that received both drugs, they were really fine. They had, we inject them 4 weeks, 2 weeks, twice a week, and we kept them another month, and they were really happy, no lost weight or whatsoever. And here, as I mentioned, 072, because it's an inflammatory Smac-mimetic, increased the spleen weight, but when we added P38, we did not exacerbate this inflammatory symptom. So that was good. So we went back to Tetralogic, and then we asked them whether we could test birinapant plus LY, because we needed that for the paper. And they say yes, and as expected, P38 inhibitor did not exacerbate the slight increase in the spleen weight. So the mice can cope both drugs. So now, do they cope this drug because we're using like low concentration, and then with this concentration, will we be able to cure mice that have leukemia? So we started by a mouse AML model that Stefan has engineered. So here he took the MLL-ENL and he added on top the luciferase gene that we can track and follow the leukemia progression using the imaging technology. So what we do is we inject the leukemic cells into the mice, and then we follow up the leukemia progression. And when we see a significant increase in the luciferase activity, we start the treatment. So in this model, we found that around day 8, we started the treatment. And the treatment was for 4 weeks, twice a week. And we monitor the mice up to 100 days. So what we found is, here it's picture of mice that had just received before the first injection, sorry. So you can see that the leukemia has taken over. And 2 weeks later, the leukemia progressed, and it's 2 weeks of treatment. So neither LY or birinapant had an effect on the leukemia burden. But when we treated the mice with the combination, we had spectacular inhibition of the luciferase activity, and which means that our combination was killing the leukemic cells throughout the body. But most importantly, what we found is we left the mice in the shelf, and we monitored them for 3 months, and we found that this combination was able to cure 80% of the mice. At least the mice were leukemia-free for 3 months, and even after we stopped the treatment. So we obtained similar results using another MLL fusion protein, the MLL-AF9, and here you can see that around 50% of the mice were able to survive leukemia thanks to the treatment of P38 and birinapant. So as I mentioned, RAS is an oncogene that is often found in MLL fusion protein leukemias. And we found that in vitro, it's not stopping the efficiency. But it was important to check whether we could see an efficacy of our treatment in mice. So here again, we are following the mice through the luciferase activity, and we found that after 2 weeks of treatment, the mice that received the combined treatment had a significant decrease in the luciferase activity compared to the one that received the single treatment. And that has been reflected by an increase in the survival in the survival of the mice, but nevertheless, they all succumbed to leukemia during the treatment. So although they were all, they died from leukemia, what we found in this model is that the combination has had a significant effect on different leukemic symptoms. So the way we monitor the mice when the AML mice are sick is we look at the white blood cells, and they're usually up, and we look at the platelet, and they're down, the spleen weight and the liver weight, because there is a lot of leukemic cells that infiltrate those organs. And you can see here that when we euthanized all the mice, we found that the combination has decreased the white blood cells, almost reaching the normal level, and similarly increased the platelet and decreased the spleen and liver weight. So that suggested that our combination was working, but for some reason the mice were succumbing to leukemia. And what I found is that when the mice were sick and received the combination, here as shown, they all have almost normal normal feature. But the only symptom that they had, it was leg paralysis. And what happened is that the cells, they accumulate into the bone marrow, and the mice, they get paralysis, and they can eat properly, and they need to go. So different option. Either there is like highly resistant cells that are in the bones and stay there, and the combination is not killing, or the drugs, they are not going to the bones properly. So this is something that we are thinking on trying to look at. So one of the most important steps in a preclinical study is to check whether the result that we got in animals can be translated into human samples. And here we're looking at the cell viability of human samples that has received either P38 birinipan or the combination. And this is a panel of 8 patients. And what we found is all the ones that were relatively resistant to birinipan has been sensitized by the addition of P38 inhibitor. So that was quite encouraging. And we are still screening more patients. And here, obviously, the number is too low. But ideally, we would like to know what would be the best responder for our combination. So to finish, I'd just like to say that we are looking for other cancer types that would be responsive for the combination. And we're screening with David Segal, we're screening cancer cell line for the sensitivity for the combined treatment. And the other 2 projects that I have is not, it's not really related to the combination, but we are looking at the responsiveness of breast cancer and ovarian cancer to birinipan. And what we found is some subtype of breast cancer, they are highly responsive to birinipan. Here, birinipan has expanded the survival of mice bearing the tumor cells from patient 322P. And here, similarly, in the patient-derived xenograph, we had a great increase in the survival of mice that receive birinipan. So this shows that they are responsive to birinipan, and it's worth considering adding the P38 inhibitor on top, just to see if we can cure the mice. So just here, it's just to finish on the take-home message is Smac-mimetic plus P38 induce TNF and increase the death in AML. And here we're showing that the association of 2 completely unrelated drug is able to treat AML, at least in a mouse model. And the good thing of this project is, although I was whinging about the number of papers, we have a panel of P38 inhibitor in front of us that we can try, and those P38 has been trialed in human, so we have all the safety profile. And similarly for Smac-mimetic, we're not married to tetralogic, so we can use another Smac-mimetic if birinipan fail in the clinic. And another option that we have, and I haven't talked much about the efficiency of MK2, but MK2 has been shown also to, we show that it's increasing the TNF and the death, and I haven't shown that, but it does also kill AML cells. And again, there is a lot of labs that are developing MK2 inhibitor, not to treat cancer, but to treat inflammatory diseases. But that's another option that we have. And another thing that is important is our combined treatment is killing cells independently on the P53 pathway, which is known to be mutated in 50% of cancer, as well as independent on RAS overexpression. So really, this work is can be directly translated into the clinic. Okay, so the best slide. As John mentioned, there is a lot of people that participate to this long but good project. And so I'm going to start by the most important before you go back to sleep. But yeah, I would like to thank John for many reasons. So the first one is taking a girl that he just met in his lab. No, it's no. It's I found that it's really brave to take someone that you don't know in your lab. That's it. So, but yeah, that's the way it is. That's the way you, but yeah. So it was really brave from him to take me in his lab. And now he's paying for it. Yeah, so for that, and for his constant support, so, and for his really constructive supervision. For the fun, we have a lot of fun in our lab. We work, but we also have fun. And the one thing that I really want to say is, I think throughout those years, is he managed to transmit to me his enthusiasm. So you saw how John is, when he presents, always enthusiastic. So I've become to be like that.
Other people, please. You've got 1 minute.
Oh, and so... Ah, putain. No. But you talk too long. That's your fault. No. And I would like to thank you to not schedule the lab meeting before 10. Thank you. Except those last 2 years. Deep, she gave me a lot of technical assistance as well as psychological assistant. So I would like to thank her for the work she's doing, but as well as for the friendship that we have. Devo, because he's Devo. And Ling-Wan has been instrumental in this study, and she was the one working on the P38 and MK2 inhibitor back in Zurich. Gabriela is also has been instrumental in the P38 story. She brought all the AML model to our division, and then we all having fun working with that. Stefan, for the multiple constructive discussion we have about the AML. Andrew Way with NUI, they are the one who tested the human samples. Ricky Johnson Labs, that is, they've been really quick at like testing the RAS model for us, so that was really handy. And I haven't presented much of the data for the breast cancer ovarian, but I would like to thank Delphine to take me on board in the breast cancer field, and Claire Scott and her army, and particularly Monique Taupe. So the drug from Tetralogic and the animal tech. Thank you.
I think we have time for a couple of questions, if anyone would like to know more.
So in one of your models, I think it was MLL-AF9 with the NRAS mutation, you had a survival advantage of 8 days from, and you said that the mice had hind leg paralysis. So is anything known about blood-brain barrier permeability for those drugs? So could there be already in the spinal cord or in the cerebellum or somewhere cells lodged rather, and you can't reach them with the drugs?
Yeah, that's the question that I'd like to address, and I don't know much about it. I know that it crossed the brain barrier, and then I'm not sure about how it goes through the spin cone and etc. We only have 8 days, but that's the best that you can find in this model. When you look at other publications, it's never over 5 days. So this is quite impressive for this really aggressive leukemia.
James.
I was just wondering if you've looked at other effects P38 inhibition might have apart from enhancing TNF production. And
Sorry?
If you've looked into whether inhibiting P38 might have other effects other than enhancing TNF production, and in regards to that, whether you've compared the co-treatment of P38 inhibition and birinapant with, say, birinapant treatment and, say, just tolerated doses of TNF or trail injection, to see which is more efficient.
So birinapant plus P38 and TNF on top.
No.
No.
So just to compare, so if you hypothesize that P38 inhibition enhances TNF to kill the cancer,
Yep.
whether it might have other effects.
Does P38 inhibitor have other effects besides inducing TNF? Have you tested?
That might be more beneficial than just, say, injecting mice with birinapant and low doses of TNF or low doses of trail. So it actually might be more beneficial than doing that.
Okay, I understand what you mean. Yeah, okay. So I haven't looked at other... Oh, yeah, no. No. This is a really good question. It was long. No. It's, I mean, if I get it. So when you do a clonogenic assay, what you find is just inhibition of P38 is inhibiting the proliferation. So is that? So the good thing is that you increase the TNF, you increase the death, and you also decrease the proliferation of cancer cells. So which probably answered your question by, yes, Smac-mimetic plus P38 is better than Smac-mimetic plus TNF. Is that? Yeah.
One last question, Rebecca.
Najoua, is the TNF receptor involved at all in the secretion of TNF, or is P38 sitting there constantly regulating open junk? Is P38 sitting there constantly regulating, or do you get P38 activation downstream of the receptor when you get activation of non-clonical antibody?
I do get an activation through the TNF1 receptor.
Completely separate on
Yeah.
TNF1?
Yeah. So when you block TNF1, nothing is happening. Yeah.
Which is difficult to explain, but
That's another.
it seems to, at least in the macrophage model, it completely wipes out the production of TNF, as does the loss of cell death. Okay, so because we listened to, because Najoua went on too long in the acknowledgement, we have run out of time, and therefore I'd like to thank you and Najoua for...